Hello and welcome. Welcome to Biology 123. My name is Renee Wolf. Make sure you put in that accent when you write my name. That's okay, just joking. I've been teaching this class for a long time. I've taught at CLC for more than probably 23 years. Not sure exactly how long. But I remember going to school as a student. And you have some teachers who seem pretty old to me. And I think, how can these teachers still teach the same class? And I have the answer. Because they enjoy it. Maybe not all teachers, but I do. I really enjoy teaching. And so welcome to my class. We'll be learning a lot of interesting things, but it is going to take a lot of work and a lot of time and a lot of organization. Our first lecture is called 1A. And 1B, the 1 stands for test 1A, A, B, C, D, E, F, G. All the way for test 1, we're going to go all the way up to G. Test 2 would be lecture 2A, 2B, 2C, 2D, etc. So first what I want to do is give you our objectives for this video. I am using a computer graphics program called GIMP, which is free. And so I'm going to be, when I want to draw a picture or write something, I will be using a GIMP board, is what I call it, G-I-M-P. So objectives. This is for 1A. You are an electromagnetic field. We're going to study a little bit about quantum physics. And look at the scientific method, which will also be in your lab. And you're going to need to know the steps in order. They're going to be a little bit different than the one in your lab. And you're going to need to know the 2 laws of thermodynamics, and we actually are going to be applying those laws for test 3. Limitations of science. We're going to look at lack of objectivity, how we really cannot be objective. Look at pseudoscience. Lab book talks a little bit about pseudoscience. Pseudo means false, so false science. And we're going to look at some of the major advances in science that are a result of unexpected discoveries. And characteristics of living things. So let me make that smaller for you so you can see it all at one time. That is my arcing electricity at the bottom of the picture. It's on my desktop. I like it. Alright, so let's get started. What we're actually going to do before we get started is I'm going to teach you a little bit about how your brain works, trying to help you understand how to study by looking at how our brain works. Because I want you all to do really well. So first we're going to do that. So we're going to get away from the GIMP board onto my notes. So what we're going to do is we are going to look at a neuron. A neuron, N-E-U, is the basic cell in your nervous system. And this is sort of what it looks like. It's made of dendrites, right here. And I'll show you another picture where they actually look kind of like trees. See right over here? They kind of look like trees. And then we have axons. Let's put right here. Axons. Those are the pathways in your brain that all the information is going to carry on, and they're going to carry it over to the dendrites. And a synapse right here, where the electrical impulses are going to be transmitted from neuron to neuron. Alright, let's see how this information is going to help you study better. So let's take a look at this. So I grew up by a swamp, wetlands, and here's a whole bunch of cattails. Now let's pretend this is like a huge, huge wetland. And there's no cars, there's no roads, nothing. And let's say there's a river like way over here and you want to get to the river. So what you have to do is make pathways. So what we would do is we would decide where we want a path, and we'd start stepping with our feet. And we lay down on it. We're just little. This actually was taller than us. Maybe our heads went up to like here. So we couldn't really see, over the top. So we would lay down, trample with our feet, lay down some more, then just going further and further, making a pathway. We had to go over and over and over the pathway. Several days, weeks. After about 2 weeks of just making sure there's nothing growing, making it wider, 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 right? Let's say you want to go to the shortest way to the river. So every day, right, we'd make sure that there's nothing growing in this pathway, keeping it clean, keeping it clear of any other type of plants that would grow. And it was very obviously a pathway. And that's what we need to do as well in our brains. So the axons, right, we have to create a pathway, and they're always going to try to find the shortest way for the impulses to travel so they can do their job. And that's where something called the hippocampus is going to come into play here. The hippocampus here, this is how it's spelled, is where everything is going to get sorted, all your information is going to get sifted, a lot of information is going to get shredded. We'll talk about that later on. And let me show you a picture of a seahorse. Take a look right there, hippocampus. Here's what it looks like. And this, anybody know what that is? That is a seahorse. Notice they look pretty similar. And that's what hippocampus means. It comes from the Greek word hippo, which means horse, and campus, which means sea monster. So we have horse sea monsters in our brains. So let's go back to why am I even telling you this, and how is this going to help you study and do well in this class and all your other classes as well. The reason why we're talking about this is because this hippocampus right here can pretty much help you not do well or help you do really, really well, depending on you. So any important information, things that you repeat over and over and over again, that will get stored in your memory. But if you listen to something, let's say if someone comes and says, oh, hello, I'm this and this, and telling your name, you're not going to really remember what their names are. And when you learn something in this class, unless you actually study it over and over and over again, you're not going to be able to remember it. The reason is this hippocampus is going to go and shred anything that your brain doesn't think is important. So it's not going to think it's important unless you give it importance. So when you get new information, what's going to happen is all that information is going to be put in the pending tray in the hippocampus. And if no attention is given to it, it's going to get shredded, maybe within hours of storage. So that's why we need to repeat things over and over and over and over again. You go over again, you're telling the hippocampus, hey, this is important, do not shred this. And by going over and over and over, what you're doing is you are making those pathways, just like we made the pathway in the swamp. You have to make pathways, wide pathways. Pathways that you're going to be traveling up and down. That information is going to be traveling up and down. The more you travel down that pathway, the faster you're going to be able to learn. Until pretty soon you're going to do it automatically. Like driving. When you first learn how to drive. Like, oh, I have to do this, I have to do this. Now you just go and drive. Or if you play any musical instruments, like piano, guitar. When you first learned, it was pretty rough. But now if you practice for a number of years, you can just sit down and play. It's become automatic. It's become part of your permanent memory. So your brain receives hundreds, billions of information every second. Billions of information. Look all around you. So much information. How many particles of dust is in the air? How many little dots on the wall? How many little fibers are in the rug? Your brain actually is picking everything up. Everything, every single thing in this room right here, my brain is picking up. Everything. But my brain is saying, hey, you know what? You don't really need to know how many stitches are in your shirt. You don't need to know how many fibers are in the rug. And it goes and shreds it. So you need to make sure that you are giving it, emphasis. You are making it aware that this information is really important. And you need to go over and over and over and over again to make those wide paths. So as you have heard probably before, if you do not use it, you will lose it. Why will you lose it? Because that hippocampus is going to shred it. By repetition, you'll have pathways, just like in the swamp. Go over it and over and over it and over and over it. No weeds will be able to grow, nothing will be able to grow. And you'll really do well. You'll be able to remember. Alright, let's get on with our lecture. So we talked about why we need to study through repetition. Because we do not want our hippocampus to shred the information that we are trying to study. One of my jobs as your instructor is to help prepare you for the future, for all these new discoveries that are being discovered. Science is changing so much. There's so many things being found. So many things that we never even dreamt of that we're finding. So this is a very exciting time. Everything is changing in science. So many things are changing. So here's just a few of those things. They're finding that there are actually more than 4 dimensions. Maybe you thought there were 3, but actually there's 4 that we are aware of. 3 spatial and 1 time. And another thing, maybe 10 dimensions. 6 are curled into less than 10 to the negative 33 centimeters. Some are inferable right now by indirect means. And they're also saying that our reality is just a hologram. The building blocks of atoms composed of neutrons, protons, and electrons. And these building blocks, which we think are solid, everything seems like it's solid, but are actually 99.99% empty space. For example, if an atom was the size of CLC, the particles within it would be about the size of a dime. If the nucleus of the atom was about the size of a golf ball, electron right over here would be about 55 miles away. This is a lot of empty space. A lot. So an atom is about 99.999999% empty space. And if you removed all that empty space from all the atoms of all the people, the entire human race could fit in the volume of a sugar cube. Well, that's a lot of empty space, don't you think? How can our world be solid? Everything seems so solid. It does. Made of atoms which are 99.99% full of empty space. By the time this table is pounding at it, sounds solid, doesn't it? But if you tell me there's nothing here, I'm like, oh, try to hurt my hand. But you say there's nothing there. You're more correct than I by a ratio of 30 million years to 1 second. The reason why it feels solid is because I'm feeling the electrical fields of my molecules colliding with the electrical fields of the molecules in the table, so I have the illusion that it's solid. It is solid in the sense that I cannot penetrate it because I cannot relieve those electrical fields, and they're working on that. And if we could relieve those electrical fields, this is what I want to do. Walk through walls. Time travel, wouldn't that be neat? All the science fiction stuff? It's all real. When I was young, I was an avid science fiction reader, so all this just seems normal to me. A lot of the science fiction stuff is based on truth. Einstein is one of my heroes of science, and so is Nikola Tesla. And they both said that everything is energy. Energy is everything. So over here, Nikola Tesla. If you wish to understand the universe, think of energy, frequency, and vibration. Albert Einstein, everything is energy. That's all there is to it. And here is one of his quotes down here. Everything in life is vibration. All that you are is energy. One way of looking at it is that solid matter, as we conventionally understand it, does not exist in the universe, for all particles are merely vibrations of energy. It's all energy, and energy vibrates. And you will be doing an assignment on energy. A lot of the nanotechnology is going to be dealing with energy, frequencies. And I've read about different types of tools and such, like building things. Instead of using all the tools that they do, they actually will use frequencies to lift up blocks, lift up wood, everything like that. They'll have frequency tools, vibration tools. Energy is the invisible power source behind all the matter in the universe. With the discovery of the electron microscope, it was discovered that the traditional model of an atom, which illustrated atoms orbiting around the nucleus of the atom like planets rotate around the sun, was not totally correct. They found that actually electrons do not orbit around the nucleus of an atom in a uniform pattern, but are rather clouds of energy flashing in every direction simultaneously. Experiments also indicate that electrons are in many places at the same time, and that an electron orbiting the nucleus is not always there in particle form. It can exist as a wave. And this is really amazing. It's like a wave until someone looks at it. And when you look at it, it becomes a particle. Pretty amazing, actually. Because how does that wave know that someone's looking at it? And we'll talk more about that in the next video. Somehow it knows that someone's looking at it, and they actually know the observer's intent. So that's something to think about there. this picture right here. This is actually what an electromagnetic microscope looks like. In 2008, they had microscopes that actually can look inside the atom. They can actually separate all the subatomic particles and see all of them. Now they have some that are even better than that. And let's pretend that our eyes, we didn't have the kind of eyes we had. Let's say the eyes were electron microscopes, and we just like, go ahead and look around the room, close your eyes, pretend like your electron microscope eyes. What you'd see was vast quantities of atoms spinning at the speed of light, each carrying an electromagnetic charge like a magnet. And by that charge, they'd create electromagnetic fields. This next part is important. Positive parts of the magnet, you probably know this, are going to repel each other. Right? Negative parts are going to repel each other. And just that. Okay? Those magnetic fields, positive, repelling positive, negative, positive attracted to negative. That makes things appear to be solid. And we are electromagnetic fields. So here they're pushing, repelling each other. So you are an electromagnetic field. I am an electromagnetic field. My material being is made of atoms with electrical charges which move at lightning speeds. They attract and repel other magnetic fields. So I am going to repel. So I'm an electrical magnetic field. That's going to repel me when I come to a surface. Which leads to some very interesting ways to look at things. We actually have a really large magnetic field around our heart area. Around our brain. So everybody has these electrical magnetic fields. They can actually take pictures of them. So let's use that information. If atoms are 99.99% empty space, what prevents objects from passing through one another? Each atom vibrates at a certain frequency according to the composition of the electrons, protons, and neutrons. Electrons surrounding the atom form a cloud that either repels or attracts other atoms with the same vibration. Now this part, the interesting part. Just think about this when you're walking, when you're sitting, doing things. You are not sitting on a chair. If you're sitting on a chair, you're not actually sitting. What you're actually doing is sitting above the chair, because the electrical magnetic field on your bottom side is being repelled by the atoms, the electromagnetic field of the chair which are pushing you up. If you walk, get up and walk, or maybe you're standing now. As you walk, you're actually walking on air. There's a cushion of atoms beneath your feet. The electrical charges of the rug or whatever surface you're on are pressing you up. So they're pressing up against the electrical charge of your feet. So just like if you put two of the same positive with positive, negative with negative, they're going to repel each other. Same thing's happening. So you're not really walking on something solid. You're walking on a cloud that you created by your frequencies. It only appears as if you're walking on the floor of the hallway or wherever you're walking. The electrons of the surface that you're walking on are repelling the electrons on your shoe, and in reality you're floating. You're floating. You're hovering over the floor of the hallway or the sidewalk. And here's another really interesting thing. Researchers suggest these clouds are ways that electrons change or blink into view from time to time and form particles of matter. Some part I might skip some of this because I always think it's going to take, never realize how long it's going to take to make these videos. But this next part is pretty interesting. So on the video that you're going to watch, you're going to see that The speed of the frequencies of the vibrations are what's really important. Like incandescent light bulb. We can't see it flickering, but it is flickering. A 60 Hertz bulb is blinking on and off, flickers 60 times a second. But it's moving so fast, it looks like one solid light. And they actually picked 60 Hertz because that's a threshold that we can't see. Our organs all have frequencies. Now look at this. We are constantly blinking in and out. Where are we when we blink out? I don't know. Like, when we go on driving and you get someone like, how did I get here? I don't remember. You like blinked out the whole time? I don't know where we go. So the difference between the physical and the invisible realm is the speed. So the speed, and I'm going to talk about one of the videos. Invisible world is invisible because it moves at higher frequencies that we are not capable of seeing. So we are limited by our senses to certain frequencies. Anything below that or above that we cannot see. Doesn't mean it's not there. Just like in this example down here, radio stations. All these different radio frequencies, all these different things. They're all in the air. But if we're not tuned in to that particular frequency, we're not going to be able to experience it. Doesn't mean it's not there. And these radio frequencies, internet frequencies, they were there since, whenever. Our electricity was always there, but we just didn't know how to access it. So what has to do with the speed, and again, you'll talk about that. But I do have to skip some things. Quantum is a big thing. Totally big thing. It's totally changing everything. I'm not going to get into all of it. We will look at a few videos for your assignments. Alright, so let's get into the scientific method. Again, this is slightly different from the one that is in your lab book. Your lab book focuses more on these first part. Doesn't actually have the complete one, and it has extra steps in between observation and hypothesis and experiment. And this is called Newtonian science, right? This is based on the scientific method. Quantum science is totally different, and beyond that we have the nanoscience. So we're learning so much new stuff. It's totally amazing. So, empirical science is, Newtonian science is what uses the scientific method. So the word science comes from Latin. It means to have knowledge or to know. And you need to know how this is different from something like history. So many fields have accumulated great volumes of knowledge but are not science. Science is limited, right? Newtonian science is limited, right, to what we can observe, what our senses can observe, do experiments on. Doesn't have to be just our senses, but extensions of our senses. So the key thing is how the knowledge is going to be acquired. So the key word there is how. And that how is using the scientific method, doing experiments on it. So scientific method, the key thing is for the scientific method is you have to have rigorous testing. Testing it over and over and over. And it's not as much scientific method as we actually really need to do, because you're not going to have enough money to test things over and over and over, and other people have to test it. And science things are constantly changing as new information is accumulated. Science has to be able to change. If it can't change when we get new information, then it's not science. Something else, it's not science. So let's look at this. So you'll need to know these steps in order. Again, as I said, this is a little bit different than the ones in your lab book. I put an asterisk by these. These is observation and experimentation are the key things in the scientific method, in Newtonian science, also called empirical science. So if you can't observe it, can't test it, it's not science. Doesn't mean it's not true. Doesn't mean that at all. Okay, it just means it's not science. So some things that you don't think science is science now, maybe in the future we will think it is science. So let's look at observation. We can use our senses or extension of our senses. Key thing here, what we observe, we have to be able to observe more than one time. It has to be capable of verification. Other people have to be able to see it. And this can change. It's changed a lot, and it will be changing in the future. The video that you're going to watch on energy is really good at this, explaining this a little bit. Hypothesis. Okay, make sure you know the difference between a hypothesis and a theory. A hypothesis is inductive. It goes from a specific observation. Okay, here we have observation. Based on this observation, the specific observation, you come up with a general statement. And it must account for, what do you think that word will be? It must account for blank the observed facts. All right? All the observed facts. And it has to be testable. If it's not testable, it's pretty useless in science. Alright, meaning this. It's over here. The average smartphone has more advanced technology than Apollo 1. The first time it went to the moon and back. Just think like in 10 years. It's going to be totally changed. Alright, experimentation. So you observe it. Alright, specific observation. You come up with a general statement about it. Trying to explain it. And then you come up with experiments. Key thing, remember? Observation, experimentation. Now, you are not trying to prove it. Science cannot prove things. It can just disprove. We'll talk more about that later in this handout. Oh, this video. It has to be repeated. What do you think? Many times. Right? Many times. And it must be repeatable by how many people? Many people. So again, you cannot ever disprove things in science. I mean, you cannot prove things in science. You can only disprove things. So you cannot observe it, cannot test it. It's not empirical science. And it doesn't mean it's not true. So if that hypothesis passes the experiments, it hasn't been disproved, it goes to the next level, which is the theory. So theory, you start out with that general statement that you made in the hypothesis, and then you apply it to other specific situations, not just the one that started out with our initial observation. Again, theories can never be proved or proven. They can only defy being disproved. Make it more and more credible the more people try to disprove it. And it's never really proven. You must be able to predict things, do more testing. It has to be open to scientific criticism from anyone. Nothing is sacred in science, which is one reason why I like Einstein. Because he would give his work to people who wanted to show that his work was false. So he wanted to find out what was wrong with his ideas so that he could try to fix them. So he was open to scientific criticism, which is what we're supposed to be. So, make an observation, ask how, what, where, when, why, devise a hypothesis, which is an educated guess, possible explanation of observation. Again, it has to account for all the observed facts, and it must be testable. Do experiments, have to be repeated many times, repeatable by many people, the more the better. Draw conclusions, come up with a theory, Then you do more testing. Other scientists are trying to disprove the theory. Again, they have to be open to scientific criticism, especially nowadays. Everything is changing, more and more information, learning so many things. And then if it survives all that, then it becomes what we call a scientific law. And we do have a few of those. Usually the scientific laws have been around for like 100 years or so. So you're going to need to know these two laws of thermodynamics. We will actually be applying them in test 3. If you've had physics or chemistry, you might have learned them a slightly different way. But in biology, we're focusing on the energy. Energy, number 1. So first law of thermodynamics, energy cannot be created nor destroyed. It just changes form. And when the energy is changed from one form to another, some of that energy is released as heat. Lost, sometimes it can be used, so maybe lost isn't the best idea. You can say released as heat. If you take physics or chemistry, you might have learned it this way. Natural processes tend to move toward a state of greater disorder. Everything goes from order to disorder. Skip that. Like hot, always going to flow to cold. Processes that are spontaneous are going to go in one direction. They're not going to go in the opposite direction. Like cold is not going to move to hot. If you have a nail right here, shiny nail, it's going to get rusty. But that's not going to just spontaneously become unrusty unless you do something. You need to do something to shine it up. But it will not spontaneously become shiny again. Law of biogenesis. Okay, know this one as well. Like produces like. Living things come from living things. Living things cannot come from non-living things. So again, like produces like. So if you are planting a garden this year and you get a pack of what says tomatoes on it, you're not going to wonder, I wonder what it's going to be. Or let's say you have some tomato plants from last year and you save the seeds. You don't say, I wonder what's going to happen. I wonder if my seeds are going to become carrots. I wonder if they're going to become flowers. No, you plant a carrot seed, expect a carrot. Plant a tomato, you get a tomato. There has been a debate, over spontaneous generation. And we know that's not true. But who knows how many things we think are true that the future are going to laugh at us. Because they had no idea about so many things. They didn't have microscopes. They didn't know anything about bacteria. They didn't know a lot of things about how frogs reproduce, etc. So people thought that toads, snakes, and mice could be born of moist soil. Flies could emerge from manure. Maggots could arise from decaying corpses. So let's say, give an example, let's say you had some meat, because they didn't know anything about refrigeration, they didn't know anything about bacteria, they didn't know about flies, things like that. So let's say you had some meat on your table, and then you notice that there's little white little wormy things growing in it. And then you're like, wow, spontaneous generation. My meat turned into the maggots. This is so amazing. You call all your friends over like, wow, come look what happened. My meat turned into a maggot. They're like, yeah, that happened at my house too. Wow. But we know it's not true, right? That's not what happened. What happened was the flies came in, laid eggs in the meat. The eggs hatched into the maggots. And they used to think that mice came from wheat, old shirts. Again, they didn't know how they all reproduced. That I don't know if you know anything about mice, but they can go anywhere. They did experiments with the mice and wheat, and they put wheat in a box, a wooden box, and didn't have any opening in it. And then they're trying to show that spontaneous generation was true. So like a week later, there are mice inside the box. But knowing about mice, I know that the mice chewed through the wood. They thought that the mice came in and they chewed a hole from the inside out. But knowing mice, they chewed a hole from the inside, from the outside in, and ate the wheat. So the wheat did not turn into the mice who chewed their way out. Or if you had any apples that ever had any worms in them, you could think it was spontaneous generation, and then the apple, part of the apple became a worm. But what really happened was, you have the apple blossom, they smell really nice. And there is a type of moth that's attracted to the fragrance. And they come over to the blossoms, and they suck up the nectar, and at the same time they lay eggs in the apple blossom. And that turns into the apple over time. And the egg hatches and eats its way out of the apple to the outside. So the apple did not become the worm, but the moth laid the egg in the blossom before it became an apple. So they had a lot of crazy ideas. Again, some of our ideas might be just as crazy to the people in the future. Like frogs, salamanders. They thought the mud, or even sometimes just the smells from a swamp, caused frogs and salamanders to turn into frogs and salamanders from the mud. That the log actually became a mushroom. Thus became fleas. So be prepared for paradigm shifts. Paradigm, a set of assumptions or concepts that constitute a way of thinking about reality. Paradigm shift, a change in the view of reality to accommodate new knowledge. Here are some of the paradigm shifts that had happened. Before microscopes, these living organisms were either plants, animals. Microscopes were developed. They discovered microscopic organisms that were neither plants or animals, they were bacteria and protists. Here's another paradigm shift. They thought that we were all born with all the brain cells that we will ever have. Shifted when it was discovered that certain areas of our brain actually continue to produce neurons throughout life. So there's actually going to be a lot more paradigm shifts in the future. So in science, when you study science, make sure you put everything like on a little shelf in your brain so you can just take it off when more information comes in. Next, we're going to look at limitations of science. Science works by falsification. So make sure you know this first one. This is also in your lab number 1. Can only show something is not true. Again, as I said, it can never prove anything. It can only disprove something. Hypothesis, after it withstands testing, becomes a theory. It's not proven, but it's not disproved. If it's not testable, can't disprove it. Doesn't mean it's not true. Doesn't mean it is true. In empirical science, non-falsifiable hypotheses are non-scientific. Cannot assert something must be true simply because it hasn't been proven false. Mathematical proofs, really not based on reality, Let's look at this one. Correlation versus cause and effect. It's important to have cause and effect. Sometimes what we think is cause and effect is just correlation. But we think it's cause and effect because we don't have enough knowledge. Here's examples. Correlation. 100% of people who breathe die. So, it would be breathing makes you die. Causation. 100% of people who don't breathe die. I guess that's true, right? Another example. Increase ice cream sales. Correlation is there's an increased murder rate. Are they related? If so, watch out for the mint chocolate chip. Perhaps there's something else that's involved here. Alright, biases or prejudices that can influence results. And that's why in science you're actually supposed to publish your work. It is so people can have your biases exposed. Preconceived ideas. And if we learn about the hippocampus, just the way you think and the information and experiences that you had will determine what is going to be shredded. So your experiences, your background beliefs will cause you to see a certain way. Unless you pay attention, alright, that this could be the case. And so if you publish your work, but it's best if you're like Einstein, give it to people who have different biases, who actually maybe don't like you, or maybe want you to fail, or a competitor, or something. Someone who's actually going to find something wrong if there is something wrong. Peer review doesn't always have the desired effect. So if you're only giving your work to people who think exactly like you, ossification, that means it's going to turn to bone, nothing's really going to be exposed. Only one certain barrier to truth, conviction that you already have it. Alright, let's look at our lack of objectivity. Objective. Expressing or dealing with facts or conditions are perceived without distortion by personal feelings, prejudices, or interpretations. That comes from the dictionary. Merriam-Webster Dictionary. We believe the visible world is real because we can access it with our senses. So if we can touch it, taste it, smell it, see it, hear it, we're going to assume that it is real. Is it? Science reports the average person, and we mentioned this before, is aware or conscious of their environment only 6 to 10 seconds every minute. Remember talking about blinking in and out? And you're only going to be aware or conscious of your environment about 6 to 10 seconds every minute. That's not very much. One reason why you got to study over and over and over again. Furthermore, although our brain processes up to 400 billion bits of information a second, we're only aware of 2,000. So our hippocampus has been very, very busy. That means we are neither conscious of, nor do we perceive most of the environment in the visible world. So even if we could judge reality with our senses, then we would have huge gaps of information missing. Going on to eat. The frequency range that our 5 senses can access is very narrow. 1% or less of the identifiable matter can be seen with the physical senses. Again, when you look at that energy video on your assignment, this will talk about that. So we can only identify 1% with our physical senses because they're very limited. That means that we cannot see about 99% of what is known to exist. So let's see. Let's skip some things. So the world is much more than we can measure with our instruments. Be aware of false assumptions. We will talk about a few of those. Scientists must be willing to challenge previous beliefs, their own and others. Again, nothing is sacred in science. Everything is open to change. Just because the majority believes something is true doesn't mean it is. The loudest is not always the correct one. You must be aware that new discoveries may force a reinterpretation of previously held beliefs. And let's look at this very famous one that turned out to be wrong. Geocentricity. The concept which states that the Earth is the center of the universe, that all other objects move around it. And this is exactly what it looks like. Versus Helio, which is the sun, says the sun is the center. Which one's true? Seems like this one, right? Get up, sun sets. The west rises in the east. Over and over again. You stand in the middle and it just goes around you and around you and around you. Here's a picture I drew for you. So this is what they used to believe. And actually people who disagreed with this, I think some of them got killed. So they believed the sun revolved around the earth in a clockwise, CW clockwise direction. Subsequently, we have learned that the earth revolves around the sun in a counterclockwise direction. So this is what we believed. Well, I didn't believe it, but Aristotle, Socrates, other people believed. There's the earth, stationary. Sun goes clockwise around it. Again, that's exactly what it seems like. If someone didn't tell us otherwise, we would believe that. But instead, this is what happens. There's the sun. We are going around the sun counterclockwise. You never heard of Copernicus? He's the one through mathematical equations came up with this. He may not have been the first, but he's the most famous. And we would spin. See, look at this axis. This axis used to be 23.5 degrees. It is no longer. We'll talk about that. So it's no longer 23.5 degrees. But it'll spin counterclockwise, and it's spinning counterclockwise as it goes counterclockwise, and it seems like this. For humors, studied Shakespeare to talk about that. Phlogiston. There's a famous phrase called "It's just a phlogiston." I actually heard it on the radio. Actually, I think it was like 2 years ago now. But it was kind of neat that I heard it. Basically, a phlogiston is an idea that everybody believes in, like the scientists believe in. But then it's disproved, and in the future, no one's ever heard of it. Alright, like this one. Everybody was studying this right over here on the left side. Geocentricity. And now most people don't even know what it is. Like, I'm sure you probably don't know what a phlogiston is. You want to know what it is? You can read it right here. Planet Earth, etc. Okay, germ theory. That's a good one to know. And before they knew about microorganisms, they know no idea about diseases spreading and such like that. Next, you know this famous fellow. Pasteur. Pasteurized milk. He had the idea that there were some kind of microorganisms that were causing disease. He actually is one of the first ones who came up with vaccines. And let's look at this guy, Lister. the father of modern antisepsis. He also had the idea that disease was caused through microorganisms. And he became the head of a hospital at this time. I forget what year it was. And most people really didn't know anything about diseases. But at the hospital, probably all the hospitals in those days, nobody washed anything. They had no idea that they needed to wash anything. And so let's say we have a doctor who goes from one patient who has this terrible disease, and then he goes and delivers a baby, and then he goes and saws off someone's leg or something, and then he goes with this, and he uses the same tools for whoever needs those tools. They're never washed. Goes from the person who's really sick, goes to other people who are not sick really, and maybe delivering a baby or need, something minor, and then many of them will die because the doctor was spreading the disease. So Lister made everybody wash their hands between patients. All their instruments had to be washed. All the blankets had to be washed. Everything had to be washed. And there was a very great decrease of people dying in his hospital. But all the doctors were really mad at him and got him ousted from the hospital. And then, once again, people were beginning to die at the same rate. Importance of hand washing, we all know that, right? This is what's most frequently missed right there. We want to focus on fingernails right there, the thumb. Frequently missed, where the orange ones are. So sing to the tune of Row, Row, Row Your Boat. Alright, pseudoscience, false science. Takes on the veneer or flavor of science. It's not supportable as valid or reliable. Lots of times the purpose is to confuse or to mislead. Maybe to get your money, your support. And it could be just the sample size is really small. Here's an example, EG right here, that means example. I use that means example. Let's say there's a commercial and everybody here in our class is going to apply for the job of being a doctor in the commercial. First of all, some people just look more like doctors than others. I know doctors who do not look at all like doctors, and I would never pick them in this commercial. First of all, it's a commercial. You don't even know if the people are doctors, and 10 is extremely small sample size. Only selected doctors might have been asked. The question might have been asked in such a way to obtain the desired answer, like, do you want this viper to, strike you and kill you? And you say no. Or do you want brand X? I want brand X. So pseudoscience, a lot of it is they don't have enough samples. They're not really what they say they are. Experts. Now, be careful of experts. Experts? How can an expert be expert? Like my brother-in-law is a not a neurosurgeon, he is a knee surgeon. His specialty is knees. He's not really good at other things because he has had to focus on that narrow field of study. For many years, that's what he's been focusing on, and he does a really, really, really good job. But look at this. Now look at this, it's doubling every 2 years. Maybe next year it'll double every 1 year. So they have all this new information. So how can an expert be keeping up on all this new information? An expert is just like us. As I say, it's you, and you're going to school 20 years. First of all, what you've learned may be out of date. And you're so busy maybe you can't keep up to date because there's so much new information. Right? And And you're an expert in one thing does not make you an expert on other things. So if you go to medical school, you're probably not going to spend a lot of time on nutrition because you have so many other things to study. But just because you're an expert in your narrow field doesn't mean you're an expert in anything else. Area of nutrition is one that is flooded with pseudoscience, is nutritional supplements. A lot of them we don't really know if they actually work. And be careful with this. Too much of a vitamin can actually be poisonous. Do not give children adult vitamins, especially not iron. And when you buy your vitamins, you need to do a little research on them because sometimes forms are sold. The ones that they sell you actually cannot actually use. And some vitamins are called bedpan bullets. You can imagine why. If they have a really, really good glue, here's just an example of a type of glue. They may be such a good glue that they stay glued together and they come out of your body intact, the same as when they went in. So vitamins are bedpan bullets, or porta potties have a lot of actually intact vitamins in them as well. And there are a lot of phytomix. You know, phyto means plant chemicals discovered today. They're unknown vitamins. We don't know how they work. They might have to work all together. If you just take one vitamin without the other ones, maybe it'd be bad, maybe they just don't work. So here's the comparison of science and pseudoscience. Science, willingness to change with new evidence. Pseudoscience, fixed ideas. Believe what you're going to believe and you don't care. Science, supposed to have ruthless peer review. No peer review in pseudoscience. Science takes account of all new discoveries. Pseudoscience selects only favorable discoveries. Science invites criticism. Pseudoscience sees criticism as conspiracy. Science, verifiable results, have to be able to do experiments. Pseudoscience, non-repeatable results. Science limits claims of usefulness. Pseudoscience, claims of widespread usefulness. Science, accurate measurement. Pseudoscience, ballpark measurement. Organic or natural? Sometimes organic are better. You have to look at other things besides just being organic. White sugar. Is white sugar natural? The reason why it's called natural is because it is derived from a natural substance, which is sugar cane. Chemical fertilizers and pesticides may have been used if it's not organic. And sugar cane is sent to refining factories where all the good stuff is taken out. Sucrose is table sugar. And then after refining, it's dried. And look at this. Pressed into crystals and it's bleached to remove its natural dark color. So if you eat sugar, you're eating bleach. It provides no nutrition. It can actually lead to bad things like dental cavities, increased cholesterol levels, heart disease, hypoglycemia, diabetes, obesity, osteoporosis. When my twin sister and I, her name is Rebecca, we went to college. We'll start back a little bit. We actually grew up really healthy. We had big gardens, we planted a lot of fruit and vegetables, so we ate a lot of natural things, and we would can our own food, can peaches, make our own jelly, things like that. So when we went to college, we were like, wow, look at all this stuff. And we ate all this stuff that had tons of sugar in it. And we became very sensitive to sugar, allergic to sugar. So we don't have sugar, we have things like stevia or agave. Honey maybe, but agave or stevia are better. Stevia is the best one for us now. And so we would just look at the ingredients and to see if it had sugar in it. But we'd eat the stuff that had no sugar in it, but we'd feel really awful. So they didn't have internet at that time. So you just couldn't look up this, other names for sugar. So we just had to go by experience. Okay, this means, okay, this. But here's some things that mean sugar, which you might not know mean sugar, in case you want to get rid of sugar because it's really bad for you. And cancer cells just thrive on sugar, and it can lead to a lot of bad things. So barley malt. Probably the ones that sugar give it away, huh? Let's look at these ones down here. If it ends in O-S-E, like maltose, that's sugar. Mannitol, sorbitol. Turbinado sugar is, okay, so we have our really super processed sugar. Turbinado is a little less processed. Ow. Sometimes you'll see that turbinado, so it's not as processed, and it looks a little brown. So you can look at these. There's a lot of other ones too. Okay, some of the major advances in science, a result of unexpected discoveries. Antibiotics. Anti means, what does anti mean? Against. Biology, life. So bacteria and fungus, right over here, these are decomposers, recyclers. They are always competing for the same food source. And they are always trying to kill each other. So the fungus produces chemicals to kill the bacteria produces chemicals to kill the fungus. So 1928, there was a man named Alexander Fleming. He was a Scottish physician and a bacteriologist. He was in his lab, and he almost tossed out some culture plates that had been contaminated. So the culture plate was open, and I drew this picture. It wasn't really these colors, I made it these colors. Let's say the red was normal bacteria, but let's say originally this whole thing is covered in red. And then, alright, fungus came and landed on that agar plate. And then what happened, you notice this, that everything around where that fungus, known as penicillium, everything around that was clear. So that penicillium, the fungus, a mold, had killed the bacteria. See, they have chemical warfare right here. He didn't really grasp the significance of this finding. Did not excite the medical profession until about 10 years later at the outbreak of World War II. So they needed effective medicine because a lot of the soldiers were dying from wounds that they shouldn't have died from, but they got infected. So they're trying to come up with some type of antibiotic. A team of researchers in Peoria, Illinois set out to coax the mold to produce more of this antibiotic. And so Fleming actually had published his work. He didn't really understand that it was really important, but he did publish his work. And we actually today still have the same penicillin. They kept it alive. So they had actually read his work, and they actually started with some of the penicillin from his actual original mold. But that was really expensive, and it didn't really work too well. It wasn't real powerful. So they appealed to the general public, asked them, please send anything that has green or bluish mold. So if you need some penicillin, that's where you're going to get it. Tell me with green or bluish mold on it. And they received huge amounts of garbage from everybody all over the United States. And they actually didn't even use any of it. One of the researchers had gone to a local grocery store, and they found a moldy cantaloupe, and they brought it back to the lab. and it was a different species of penicillium than Fleming had used, and this particular one produced 25 times as much penicillin. Then they're able to isolate a strain that produced more than 80 times the original quantity of penicillin. So they actually began to produce penicillin that worked, and that was during World War II. Since then, they've discovered more than 4,000 antibiotics, substance producing a living organism that interferes with the normal metabolism of another living organism. So antibiotic against life, bacteria and fungus, chemical warfare. So from Penicillium mold, often seen growing on the surface of rotting citrus fruits, produce at more than $1 billion a year. If you like smelly cheeses, blue cheese, alright, that's penicillin. So penicillin antibiotic produced by a fungus, Penicillium. Another very important thing that was found, not through trying to find it, but by accident, was nylon. He was working with polymeric esters or polyesters. Didn't really find them very useful. And one day an assistant, whose name is unknown, dipped a glass rod into a molten mass of the polymer that he was working with, drew out some of the material, stuck to the rod, and then it stretched, formed long filaments. Even after it was cold, the strand could be pulled by hand several times its original length. Unlike rubber, it didn't spring back to its original length, and it wasn't real strong. So that, like, you realize something really happened, something really important. This happened. And they've done experiments, and now we have nylon. Microwaves also found by accident. Microwaves used for cooking. This is the story. Man was working in the military at a base. They're sending out microwaves out into outer space to see what was going to come back. This guy had a candy bar in his pocket, got in the way of the microwaves, noticed that the candy bar in his pocket melted, thinking, okay, microwaves, melted candy bar, use microwaves for cooking. Alright, let's look at this. Characteristics of living things. Organization. We're going to be focusing on cells. And P will focus on tissues and organs. And we're actually going to look at populations a little bit at the end. All living cells are composed of basic units called cells, and we're going to spend time on cells. They can range from 1 cell to trillions. Average human body contains approximately 37.2 trillion cells. So organization. Another characteristic is irritability. Not irritable like you might think, ornery, but able to respond to the environment. To maintain homeostasis means the same environment. Like we need to maintain homeostasis in our body, like 98.6 Fahrenheit. So they respond to the environment. Movement. Living things can move. We'll look at cilia and flagella in the paramecium amoebas. Those are protists. Plants. Let's look at plants. Okay, I drew you a picture of a flower. Hold on. Alright, let's say this is a sunflower. They actually will be able to move, not, like getting up and jumping around. So let's take a look. Here's the sun. Oop, that's not have a green sun on it. Alright, so here is our sunflower. And we know that sunflowers contract the sun, so here is our sun. What? Sorry about that. Just hit the wrong button and it magnified it by 4,000 times. So here's a new picture. Alright, so let's say this is a flower. And it's going to be tracking the sun. So first of all, let's say the sun is over here. It is going to hit the stem, and on the opposite side over here, it's going to be releasing some hormones, growth hormones. So that means this side over here is going to grow longer. So that side will grow longer. So now that will lead toward that way. So initially it was this way, then the sun moved over here. So when it was over here, there were released growth factors on this side, causing that side to grow longer than this side, so it went over toward this side. The sun is moving, now it's over here. It's going to release growth factors on this side over here, so now this side is going to grow longer. So the side opposite, so here the lighter green over here, this is going to grow longer than this side because growth hormones are being released on the side opposite of the sun. So that's going to bend over that way. So it's going to be following, moving towards the sun. Another one is metabolism. Everything's obtain and use energy. We're actually going to spend a lot of time on energy in test 3. We're going to be looking at the flow of energy. We're going to look at something called photosynthesis, trapping energy from the sun, converting the solar energy into a form of energy called ATP. And that's going to be in cellular respiration. We're going to study mitosis, how things can grow and develop. And reproduction, we're going to look at something called meiosis right here, meiosis. So mitosis. That's going to be on test 3B. And we're going to be looking at adaptation and homeostasis. So let's look all of them together and then we'll be done. Our first video. So characteristics of living thing, organization, irritability, able to move, metabolism, using energy, growing and developing, mitosis, reproduction, meiosis, adaptation, and we're going to be looking at genetics. And that's it for our first video. Feel free to stop the videos anytime. You don't need to watch them all at the same sitting. But remember, your hippocampus, don't let it get all shredded. Take notes, that helps if you take notes. Make sure you look at the video questions while you're watching the video. That will be really helpful. Alright, until next time, be well.